Yeast is an organism responsible for different industrial processes such as drug development, research, and food production. It may be used in the creation of antifungal drugs, incorporation and research for cell death, and production of bread and chocolate. This organism is also a main component in bioethanol production, where yeast is used to form ethanol through a process called fermentation. In fermentation and bioethanol production, the type and source of the yeast used may affect the fragrance, flavor, and taste of the product. In our surroundings, yeast may be obtained from different sources such as nutrient-rich environments. An example of a common source of yeast mentioned in published studies are flowers because of the nutrients it can provide for yeast growth. Several research showed that yeast grains collected from flowers may present different characteristics in terms of its bioethanol producing activity, depending on the type of flower it was collected. Good day, everyone. I, Jazmine Maxene Ruba, together with my research mates, PJ Rich Liza and Jamir Jibreel Usman, and our advisor, Ma'am P.D. Capcut, present the significance of the study, isolation and characterization of yeast from selected flowers in PSHS SRP with potential bioethanol producing activity. And we are from Philippine Science High School, Soxargen Region Campus. The Philippines is known to be a very diverse country. Data shows that flowers in the Philippines reflect this very characteristic. It was mentioned that yeasts may inhabit flowers and the source of yeast may affect the product quality during fermentation. Many studies were already conducted to characterize the bioethanol producing activity of isolated yeast from flowers, but these are focused outside the Philippines.
This research aims to evaluate the ability of flowers as sources of yeast through isolation and culture, and assess and characterize the isolated yeast for its bioethanol producing activity through fermenting glucose and sucrose. The flowers in question are Gumamela, Alamanda, Bamboo Orchid, and Bougainvillea. Then, the suspected yeast colonies shall be subjected to microscopic examination for the identification of yeast cells. Samples were collected within PSHS SRC. These samples were then submerged in a liquid YEPD media and were left to incubate for 7 days. Meanwhile, a differential solid YEPD media was also prepared for the microbial culture of yeast. After 7 days, aliquots from the submerged samples were streaked onto the differential solid YEPD media. This culture was then left to incubate for another 7 days to encourage the growth of yeast. After the incubation period, creamy, glossy, and pearlescent colonies are expected to form, which were subjected to microscopic examination for the identification of yeast cells. Finally, the same colonies were then subjected to fermentation of glucose and sucrose. Inoculum of yeast were diluted and was added to the sugar solution. Bioethanol was expected to form from this treatment after 30 minutes. An iodoform reaction was used to indicate the presence of ethanol. The formation of yellow precipitate indicates that the iodoform reaction took place, indicating the presence of bioethanol.
Analyzing the ability and potential of flowers as sources of yeast and characterizing their bioethanol producing activity can contribute to the scientific knowledge and scientific community. Here are the significance of the study we conducted regarding characterizing yeast isolated from flowers. First, baseline information. This study can provide baseline information regarding the ability of flowers as sources of yeast and the characterization of these isolated yeast in producing bioethanol. Second, increased utilization of flowers. This study could also lead to the increased utilization of flowers in the scientific and economic fields. And third, economic and scientific development. This has the potential to advance economic and scientific development in the Philippines and could also promote biodiversity conservation. This study made use of different concepts in STEM, specifically in science. These are, but not limited to, the following concepts. First, fermentation. Fermentation is the concept used and involved in characterizing the isolated yeast for their bioethanol producing activity. Second, microbial culture. Since our study involved microorganisms, it is important to have knowledge about microbial culture. This allowed us to have a successful isolation of yeast from flowers. Third, laboratory techniques. This study used different laboratory techniques such as plate treating, sterilization, aseptic techniques, mixing of solutions, and microscopic examination. And lastly, the formulation of the growth media. One of the major parts of our methodology is the formulation of the growth media, as the growth media will serve as the environment for the isolated yeast to thrive. This study used the YEPD media, or the yeast extract, peptone, and dextrose media. And that will be all for our study. I hope you have learned something about yeast isolation and characterization of yeast through fermentation. Once again, we are from the Soxargen region at Mahusay.